We're now entering into Chapter 6. We're entering the second major portion of this class. There are 4 total sections. This chapter is concerned with microbial nutrition and growth. Like all life, there are needs that must be met in order to sustain living cells. Here, on this slide, are some terms and concepts to think about regarding growing microorganisms in the lab. Let's go over a few definitions. First, the term microbial growth. This term is used in a way that is slightly different from the way that we refer to other organisms growing. If you think of a human growing, you imagine the person growing larger. That's not the case in the microbial world. For microbes, an increase in the size of the population, rather than the increase in the size of an individual cell, is what is being conveyed. So for microbial growth, we are talking about more microorganisms, the population getting larger. Microbial growth equates to more cells. A colony is something that we can actually see forming on a petri dish. A colony is a group of cells, an aggregation of cells, that hopefully are the descendants of a single cell that continue to replicate until a visible clump appeared on the surface of the medium. Ideally, a colony is a group of cloned organisms. They're all the same. They're genetically identical. A biofilm is a term that we've talked about before. This is a collection of microbes that live on a surface. Typically in nature, it's a complex community. There are different types of microorganisms growing within a biofilm that forms in the environment. Notice the image that is colorized to look attractive. There are different types of microorganisms in this group. They live in a community and they have relationships with their neighbors. The growth requirements of an organism are similar to other living cells. Microorganisms need a variety of different nutrients to meet their energy needs and to have the necessary components to be able to build the molecules they need for their own cellular structures. Most common nutrients contain the necessary elements such as carbon, oxygen, nitrogen, and hydrogen. But there are other types of nutrients that microbes need in lesser amounts to maintain their structures. More on these requirements shortly. If we look at this table from your chapter, it divides microorganisms into 4 major groups. You can understand these groupings if you understand the energy source and the carbon source. Across the top of this table, we have 2 different types of energy sources. It's either light, which is designated by the prefix photo, or the energy source is chemical compounds, which is designated by the prefix chemo. The carbon sources also come in 2 varieties. Carbon may be supplied by either carbon dioxide, which is designated by the suffix auto, A-U-T-O, or carbon is supplied from organic compounds, which is designated hetero. For example, if you have an organism that uses light as its energy source and carbon dioxide as its carbon source, it would be a photoautotroph. Like the cyanobacteria and algae we talked about in the last chapter, the microbes that photosynthesized. Interestingly, there is another category of organisms you might not have realized existed until now. The microbial world is so diverse. There are microorganisms that use light as an energy source, but they can't use carbon dioxide as the carbon source. They need organic compounds. These organisms would be termed photoheterotrophs. Similarly, if we're describing organisms that use chemical compounds as their energy source and carbon dioxide as their carbon source, they're termed chemoautotrophs. These organisms are ones you might find at the bottom of an ocean near the sulfur vents and other unique environments. Lastly, we can describe organisms that are similar to ourselves, organisms that use chemical compounds like glucose for the energy source and also use organic compounds for their carbon source. These are chemoheterotrophs. We belong to that group, as do other animals. These four terms describe the four major groups of microorganisms in relationship to the growth requirements for energy and carbon. Oxygen requirements do vary in the microbial world. Not all organisms require oxygen, and some require only a little. There are quite a few variations for the requirements in relation to the presence or absence of oxygen gas in the environment. Obligate aerobes do need oxygen. These organisms use it as their final electron acceptor in the electron transport chain. They use cellular respiration. However, oxygen is deadly for obligate anaerobes. These organisms have to exist in the complete absence of oxygen because they don't have the enzymes capable of disposing the toxic forms of oxygen. Oxygen is a strong oxidizer, and the chemical reactions within the cell can generate toxic forms of oxygens that need to be addressed in order to prevent damage to key cellular molecules. This slide is showing the definitions of the different types of microorganisms as it pertains to their oxygen requirement. As mentioned, aerobes, strict aerobes, require oxygen to be present, and they use aerobic respiration. Strict anaerobes do not use aerobic metabolism, and they actually require the absence of oxygen. Facultative anaerobes can maintain their systems in either condition, oxygenated or anoxic, because they prefer oxygen but they can live without it. They are designated facultative anaerobes. A lot of the organisms that live in our gut are these types of organisms. Think of bacteria like E. coli. I'm sure you've heard that name before. E. coli falls in the category of a facultative anaerobe. Our gut is anoxic, no oxygen gas is present. Aerotolerant anaerobes have metabolic systems that are not using aerobic metabolism, but they do have the ability to dispense with some of the toxic forms of oxygen. Therefore, they can tolerate the presence of oxygen. Microaerophiles are organisms that exist in a slightly oxygenated environment. They have only a limited ability to detoxify molecules like hydrogen peroxide and the very toxic superoxide radical that forms in the presence of oxygen. These organisms require a low level of oxygen, but they cannot exist if there is a high concentration or a normal atmospheric concentration of oxygen. Many different nutrients cycle through the biosphere. This is just a single example, but it's an important one. This slide is illustrating the nitrogen cycle. The nitrogen atom is required in several important biomolecules. Remember the NCC pattern of amino acids? Proteins need nitrogen. Also the nitrogenous bases, the G, A, C, T of DNA, or GAUC in RNA, have nitrogen as part of the molecules. Nucleic acids need nitrogen. Nitrogen is a key component of life, but it's often a limiting factor. This is because while there's a lot of oxygen in because while there's a lot of the atom nitrogen around, it exists in a form that is not available to most life. If you think about nitrogen gas, N2, the form that exists in the atmosphere, there's a lot of it, but N2 has a triple covalent bond. It can't be cleaved apart by very many organisms. You might not be surprised to hear there are a few types of microorganisms that can break that molecule apart. Those are called nitrogen-fixing bacteria. You'll notice two spots on this cycle. Here, coming from the atmosphere into the soil, over there on the left, and then here from the atmosphere down into the aquatic environment to the cyanobacteria over on the right. In these two spots, there are examples of bacterial species that are nitrogen fixers. These species can break apart nitrogen gas into forms that are usable, forms like ammonium that you see here. Those useful forms of Nitrogen-containing molecules can cycle through the aquatic or the soil environment to support cells. Some of it will eventually get returned to the atmosphere by yet more microorganisms, the denitrifiers. These other circled types of organisms in the soil and the aquatic environment are the denitrifiers, and they take molecules like nitrate and turn it back to nitrogen gas, which returns to the atmosphere. This is the cycle that involves the atom nitrogen. A similar type of cycling happens for all of the different important molecules and atoms. Microorganisms are always a part of that cycling. Keep in mind that it isn't just those 4 key atoms, carbon, hydrogen, oxygen, nitrogen, that cycle. There are other requirements for life. Phosphorus in the phosphate groups is also really important. It's key to the phospholipid bilayers of cell membranes. Phosphate's also a part of DNA, RNA, ATP. Sulfur is an important component for certain proteins. Amino acids like cysteine contain sulfur, and sulfur-containing amino acids are important for forming covalent bonds to hold the 3-dimensional structure of proteins together. Yet other elements are required in trace amounts. Very small amounts of some atoms are needed to power enzymes in cells. The amount required is so little, sometimes there's enough dissolved in just tap water to satisfy the requirements. But all these elements are required and would be involved in nutrient cycles. However, some organisms can be very finicky. They may require some specific growth factors, specific organic chemicals that might be found only in, say, a host animal. The organism can't synthesize it themselves, so they need a molecule provided to them in order to grow, so they can be more difficult to grow in a laboratory situation. In regards to physical requirements, these too must be controlled in order to maintain your organisms. Microbial cells have specific needs just like you do. For example, they can only exist in specific temperature ranges. That's because their proteins are affected by the temperature. If a cell gets too hot, proteins will denature. The activity of the proteins will cease and the cell will die. The temperature graph on this slide, it has somewhat a skewed bell-shaped curve. They might grow, albeit slowly, at low temperatures. They multiply faster as they approach an optimum. And if they're placed in a temperature that's very far beyond the optimum, the growth crashes. This is because of the effect on the proteins. What is temperature doing to the protein? As I mentioned a moment ago, if it's too hot, it'll denature the proteins. If it's too low, membranes become more rigid and inflexible, and proteins stiffen as well. Membranes and proteins can become very rigid at low temperatures, and then much more loosey-goosey and fluid at high temperatures. At high temperatures, membranes lose their integrity. The cell will die as it can't maintain homeostasis. Because there are different temperature ideals for different types of microorganisms, there are terms applied to these different temperature ranges. The phile, Phile part of the term, means liking. So a hyperthermophile, shown in the red graph, likes it really hot. There are organisms that might exist in the boiling hot springs of Yellowstone or some other type of thermal vent in the ocean or hot spring. Thermophiles like it pretty warm, hotter than our own body temperatures. They prefer temperatures around 60 degrees Celsius. The mesophiles are happy at the temperature that's close to our own body temperature. They're called mesophiles. All pathogens and disease-causing organisms are going to exist in this range because they're causing problems in our bodies, and this is our temperature. The psychrophiles like it cold. Here, psychrophiles belong in that bell-shaped curve towards the end. They like it cold. There's one more term that you should be familiar with that's missing from this slide. It's a term for organisms that exist as a type of in-between the psychrophiles and mesophiles. I'll throw that term out here for the first time, but I'll be using it again later in the semester. The term is psychrophile. No, psychotroph. TROPH. Psychotrophs actually have an ideal growth temperature that's in the mesophilic range, but they also have the capability to grow well into the psychrophile temperature range. For example, we have a pathogen called Listeria monocytogenes that can cause problems in food because it's a psychotroph. Therefore, if it ends up in your food, which you put in the refrigerator, it will happily multiply in the low temperatures of the refrigerator. Later, when you ingest it, the numbers are quite high and it can make you sick. It causes a type of meningitis, especially in the fetus of a pregnant woman, but also in immunocompromised people and the elderly as well. These high-risk individuals can get quite sick from this particular organism. Listeria monocytogenes is a psychotroph. Their optimum growth range is in the mesophilic range, but they can grow quite happily in the psychrophile range. Next, pH requirements are specific for organisms. Organisms also have a very specific range that they can exist in. That's because the hydrogen and the hydroxyl ions will interfere with the hydrogen bonding in proteins and nucleic acids. If the pH range shifts too far out of the optimal range for that organism, then you'll find that the hydrogen bonding capacity is not ideal in these various larger molecules, and that affects function. Organisms prefer the specific range for which they are adapted. The neutrophiles like a pH close to neutral. That's where we would exist if we were a microorganism. Our enzymes, for the most part, are happiest in the pH range closest to neutral, except if you're looking at enzymes that exist within specific compartments like our stomach. Our stomach has a very low pH, and the enzymes that function there would function in an acidic pH range, much like the enzymes of acidophile, which are the organisms that grow best in acidic habitats. When the pH is low, acidophiles grow best. The alkalinophiles like high pH ranges. They like alkaline environments. Again, the point here is that the microbial universe is really diverse. You can think of an environment and there will be microbes occupying that space. Lots and lots of different niches exist within an environment, and different microorganisms will have adapted to exist within them. But water is a universal requirement. All life needs it. So as long as there is water available, life can exist. It's important to remember water is the solvent. The enzymes and the nutrients must be dissolved in that aqueous environment inside the cell. It takes part in many of the metabolic reactions. During the synthesis reactions, water is released, and then during the hydrolysis, the cleavage reactions, hydrogen and hydroxyl is replaced by splitting water. Cells, for the most part, will die if there is a lack of water. However, some cells do have the ability to persist as endospores. In Chapter 3, endospores were introduced. Some gram-positive bacteria can form endospores, and that protects them. An endospore is essentially a dormant or hibernating form of the organism that will remain viable for years until the environment becomes favorable again. Endospores in a favorable environment will germinate and start growing again. Cysts are a form that some protozoa, like Giardia for example, will take to resist environmental pressures. They too are very hardy and can resist drying. Some protozoa make infectious cysts that will remain dormant until they become ingested by an organism. Osmotic pressure, that concept of water moving across a cell membrane. Cells need a way of dealing with changes in osmotic pressure. Some cell types are very sensitive. If you move the cell outside of a very tight range of osmotic pressure, they will not tolerate it and they'll die. If they're in a hypotonic solution, water will move into the cell and the cell will swell up and it might break open. Hypertonic environments will pull water out of the cell. That might prevent the cell from existing because the internal solution will become too concentrated with ions and other substances. If you think about how we preserve some foods by salting them or adding a very high sugar content like with jams and jellies, it works because that hypertonic environment will pull water out of the cell. There are types of microorganisms that like very high osmotic pressure environments. Obligate halophiles like salty environments, and they'll grow well up to 30% salt, whereas facultative halophiles will at least tolerate high salt. If you put a facultative halophile in a very high salty environment, they won't die. They might not thrive, but they'll survive. On this slide is a photograph of salt ponds. In areas where they make sea salt, they let seawater into these ponds and they basically just let the water molecules evaporate out over time. The ponds become more and more salty. You can see salt forming along the edge of these different ponds. The different colors of these ponds are actually representing changes in the concentration of various types of halophilic microorganisms. As the salty water is becoming more and more salty, different microorganisms are becoming dominant in those ponds. The halophiles have pigments. The pigments of these halophiles color the water of the ponds. There are some of these salt ponds south of us in Imperial Beach, California, near the border. I've seen these ponds shifting to different shades of green and orange colors over time. And there are huge piles of salt next to the ponds where they're processing the salt, mostly for use in animal salt licks. You know those big blocks for animals to lick like cattle or horses on a farm? They make those animal products with salt from that facility. This is a good point to stop, and we'll pick up with the second half of Chapter 6 in the next presentation.